I am going to start with our first question from Zoom, which is around journals. So I love this question around scientific literature. I've been an open access advocate for a decade or more. So the question is, do you think open source journals should be more accessible to the public? Why or why not? And so I think maybe there are two certain questions here. So there's this open source stuff like biorXiv, medRxiv, and the OG archive, which is like the math and physics open source preprint server.
Ooh, right.
And then and that's been around for a really long time, by the way. So math and physics people have had it going on for way longer than biologists. So props to them. But the peer-reviewed stuff is really inaccessible. So what I would love to hear, like everybody here on our panel, like where are you with that? Do you think that this should be open to the public?
So open to the public as in like accessible to read.
Not paywalled, I guess.
I think that's the main thing. That's at least how I understood it. And I'm like Jeannie, a very big advocate of open source, and I have been for a very long time. But I'm interested to see what Emily and Lindsey think.
Okay, for that, absolutely yes. I wanted to clarify whether it was like accessible without a paywall or accessible in the way that it's written, because I mean, I think too, like to some degree that could also be made a bit better in terms of like the clarity with which we write about our research. But definitely having the research at least free, like, you know, no price, free, accessible to the public should absolutely be the case, in my opinion. Yeah.
Yeah, I agree completely with everything that Em just said. So.
That's great. Yeah, I mean, I think it makes complete sense. A lot of this research is publicly funded, so it makes complete sense for the public to actually have access to the results, the peer-reviewed results of this science. So not just the archives, but actually open source publications like PLOS and others would be
Yeah, there's a couple
really
of
great
open source journals like eLife, PLOS.
eLife, yeah.
And I will say that all of the COVID-19 stuff is open to everybody on most journals. I believe that this is open.
Yeah, Nature and Science for sure.
Yeah, great.
Yeah, nice.
Okay, well, oh, sorry, did you? I think
to
I'm gonna.
Or maybe Pedro, you can take the next question.
Okay, cool. Yeah, that's what I was gonna do. Sorry, I think I got a little bit of audio interference, but yeah, I think this is a good question from Amanda, and it goes into the infectivity methodology that you were talking about. So she wants to know if these drugs were good on cells already infected, the drugs that did work on cells that were already infected, or on cells before they were infected by the virus.
Yeah, so in this paper and in the figure that I talked about, they treated the cells with the drug prior to infection, so kind of like prophylactically, sort of. And so they also, in this paper, we didn't talk about it, they looked and they saw that the drug was pretty good at preventing infection for if you gave it up to 4 hours after you infected the cells. And so what that really means in terms of in an organism, like, I really don't know, maybe someone else knows better. But it's really hard to say what 4 hours means in a cell in terms of like the timeline. And I think that's a really important follow-up question, because obviously we need to find drugs that can be used to treat patients who already have SARS-CoV-2. And prophylactic is great too, but.
Great. So we have this wonderful question from Facebook from Anissa, and you did not talk about this explicitly, but if you caught it in the methods, like how do they know if they prevented or reduced infections with these drugs in the cell culture experiments?
So basically the way they did it was, I think this might have been part of the idea behind designing it, where they gave the drug before the virus, is they basically like gave different cells like plus minus drug the same quantity of virus and saw like, in theory, like if the drug does nothing, you should see the same degree of infection in both conditions. But if the drug is preventing infection, then you would see less infection in the cells that receive the drug, if that answers the question. But that was kind of how they sought to measure that difference in infectivity.
How do they look at infectivity? Like how do they measure if something is more infected or less infected? Were you able to gather that from the methods?
I believe it was based off of just the amount of viral proteins or the amount of viral RNA within the cell. I believe that's what they did.
Interesting. Cool. Thank you.
All right, so we've got another question here from Joanne, and they want to know what type of cell lines were used for these studies. And I think you mentioned that at some point if they were lung, cardiac, or neural, and they're referring to specifically the amounts of the ACE2 receptor, which we already know is involved in the virus entering the cells, that exists in cells from these different types of tissue. And they mentioned human cell lines. I remember you guys talked about monkey cell lines at some point, but I don't know if you want to go into that in a little bit more detail.
Sure. So for the original viral protein human interaction experiments that we saw at the very beginning, those took place in HEC293T cell lines. And those are kind of like a workhorse cell line that we use for a lot of stuff in cell biology. Even just kind of like piloting and testing like certain like gene editing experiments. Like a lot of times we do them in 293Ts just because they're super easy to work with. So those are originally derived from human kidney cells. Or human kidney, I should say. And then for the later experiments, the infectivity experiments, those were in, I think they were Vero-6, if I'm remembering correctly. Good.
Mm-hmm.
And those are monkey kidney cell lines. And we use those, from what I read, particularly in virology, because they're fairly easy to infect and also like monitor infection in pretty easily. So like they kind of design their cell lines based on sort of like easily designing their experiments and then getting very clean results they can control for.
Great. And I think this is a question that maybe I could be the one to answer, because it's around how do students get involved in biomedical research during this time? And as the director of RockEDU, who runs biomedical research programs for students, I can tell you that I don't know right now. It's been a really tough time for the research landscape. I know a lot of summer programs are being canceled. I know for us, our summer science research program is going virtual. And so we are currently dreaming up ways that we can provide research experience to our students and beyond, so that they can go through the process of science themselves. And I just want to point out that in terms of the process of science, what we really want to see people doing is this like iterative process. You're going to design something, you're probably going to have some aspects of it that fail. You're going to figure out how that failed, fix it up, and then redo it until you get an answer that feels rigorous and can be replicated by someone else in an independent setting, right? And so that's the key to science, that it really needs to be something that can be replicated. And stay tuned for our D4P section. We're hopefully going to have a little bit more stuff coming soon in the corner store science section that will allow people to engage with the process of science, and we're developing out a couple of model systems that you could do at home. And so stay tuned for that, and hopefully we'll have a better answer for you in the coming weeks for how to get involved.
Great, okay, then we'll move on to the next question, which is from Samantha. And Samantha wants to know I think probably directed actually at the two of you specifically, how long have you been following the trials? I'm guessing how long have you been looking at clinical trials and using all of the tools that you showed us in the end that were super useful?
Well, not that long. So when I used to work in pharma, I paid like a little bit more attention to them, and that's how I got familiar with the process generally, just from like kind of being more in that world and in that field. But kind of like, I think as everyone, has, with all the news that's going on and like kind of the quick pace of like, oh, like this drug seems to be working. Now this drug seems to be working. Now we're testing this drug. Like I've definitely sort of tuned back into it again, just to try and keep tabs on everything that's going on. But even still, it's very hard.
Yeah.
But,
I loved your sort of reminder at how all the different phases work. I thought that was really, really useful.
Oh, glad to hear it. Great.
So I'm going to ask a question from Lauren, and I love this question. So considering that the study found that SARS-CoV-2 interacted with 332 proteins, so just clarifying that certain proteins within the SARS-CoV-2, proteome interacted with 332 human proteins. And so she asked if a drug would be needed to prevent all of these, do you have to make a drug to prevent all of these protein-protein interactions or just a few?
Yeah. Well, so I think we'd still need to like figure that out for sure, but I doubt that would be the case. If anything, I guess an example of this, I guess that we could cite maybe like tentatively from the study is that some of the drugs that just targeted like one or a few interactions did reduce viral infectivity. So that already suggests that we don't need to go after all of these proteins, which would be a doozy. So that's good to actually see some effects. And I think sort of what's maybe more helpful or like could be more informative to us to pay attention to here is kind of like what particular functions, like we showed some of those interaction maps that show proteins that are all involved in the same job. Like if certain drugs that go after a certain function are really effective at preventing infectivity or reducing infectivity, then it might just be those functions and then proteins involved in that function. That would be important for us to target. Another caveat to this too is, as Lindsey mentioned at the beginning, the way that they sort of did these experiments is they expressed one viral protein at a time in these human cell lines. And that's not how like viral replication actually happens, because a virus gets in, it replicates its entire self all at once. So you'd have a lot of viral proteins in a cell at a time. So it's possible that like in a condition where you have just one viral protein bopping around, you might have interactions happening there that you wouldn't necessarily see, as opposed to normal infection where you have all the viral proteins in the cell at once. So this is the kind of thing that would have to be tested sort of empirically, but my guess would be probably not, which is good.
And I think it's worth adding that most drugs that are small molecules, so most of, like the stuff that the pills that you take are usually a small molecule, and a small molecule will typically only have a single target, right? In just generally speaking. And so if they can disrupt one of those targets, they don't have to disrupt everything else, because that's what we're just looking for. So yeah, I think it's really nice to think about in terms of the science. So cool.
Okay, so another question from Magnolia now, and she stresses it's a hypothetical question, but I still think it's a good question and good to go into about the FDA approval. So she asks, if an existing drug was found to help treat COVID-19 and was FDA approved again, so for that purpose, what effect could that have on the people who already use that drug to help treat other conditions? Is there a way to produce medications on a larger scale, or are there restrictions?
Yeah, it's great. Lindsey, do you want to take this one?
Yeah, I guess I'll start. My real answer is I don't know what kind of an effect it would have. I know it's been pretty, like, in addition to the news talking a lot about how chloroquine and hydroxychloroquine maybe might treat COVID-19, there's also been a massive shortage in those drugs for people who actually need them to treat other things like rheumatoid arthritis or lupus, and that's a really big problem. So I don't know the answer to how can we if we find a drug that works, how do we mass produce it? But I do think that's a super important question to ask.
Mm-hmm. That was my favorite niece, Maggie. So thank you so much for your wonderful question. Okay. Here's a question from Adri. I hope I'm pronouncing your name correctly. What evidence do they have to determine if the drug has effectively bound to either that human protein or to the viral protein to disrupt that protein-protein interaction?
Okay, so for human proteins, generally so the sort of like first way that people will kind of validate drugs to see if they bind is basically they'll look for a drug that disrupts if it's an enzyme, like a certain reaction the enzyme participates in, or it'll look for just the separate proteins that are both involved in the interaction and see if the drug is there, does this enzymatic reaction not happen? And that would suggest the drug is preventing it from happening. Or these two proteins that we can usually isolate in the way that Lindsey described, while they're stuck to each other, are they no longer stuck to each other when we give the drug? So there are sorts of that's kind of the initial pass. But another way that people do it is they can actually, and this I'm not super familiar with, but I've just seen figures of it before. We can do this technique called X-ray crystallography that actually lets us see physically the protein and the compound bound together and sort of verify those chemical interactions where the compound, or the molecule, I should say, is kind of like basically like filling that binding pocket in the protein, like that actually lets us see that it's truly happening. But otherwise it's more kind of like a functional sort of like, how does this protein like do different things as we would expect it to if the drug was binding to it.
Okay, so I think this question goes back to what we talked about a few minutes ago, actually, about giving the drug before or after the cells were infected with the virus, but I'll still let you expand. Do you think that some of these drugs at some point will be used prophylactically for the general public who is not yet infected to prevent infection?
Maybe. I think that it depends a lot on like what other side effects they have. Yeah. So that's always something that is kind of that's to keep in mind with regards to kind of like giving drugs for certain indications. There's always like a risk to benefit ratio that you have to consider, where especially if it's a prophylactic, if that drug comes with some pretty harsh side effects, then you might not want to be taking it just on the off chance that you might get an infection. But if you already have the infection and it's really bad and there's nothing else, then you kind of are more willing to kind of stomach the side effects if it'll help you get better. So I think like that could come into play in terms of those decisions, but all of that stuff has to be tested in trials. So
absolutely.
Yeah.